Hello everyone, this is Priya here. I'm here to represent my topic microfiltration and ultrafiltration. So, first, let's discuss what is filtration. The separation of solid particles from the liquid and gas. So, filtration you have seen in our home, like making tea. After making tea, we filter the tea, right? So, this is filtration. So here we will discuss first some terms related to the filtration. Feed. The solution to be concentrated or fractionated. Permeate. The filtrate, the liquid passing through the membrane. Retentate. The concentrate, the retained liquid. Flux. The rate of extraction of permeate measured in liquid per square meter of membrane surface area per hour. Concentration factor. The volume reduction achieved by concentration. That means the ratio of initial volume to of feed to the final volume of concentrate or retentate. Membrane fouling. Deposition and accumulation of feed components on the membrane surface or within the pores of the membrane cause an irreversible flux decline during processing. So you can see from this diagram the feed flow. We have put the feed flow in the funnel, or we have used a filter paper over it. So when we put the feed flow, the filtrate will go down and the contaminants or the undesired particles or the substance which we want to separate retained over the filter paper, and this is called retentate, precipitate. So, filtration is an effective method for the sterilization of heat sensitive material. It acts as a barrier and remove the contaminating microorganism from the desired liquid or gas rather than destroying them. Some example of heat sensitive materials are antibiotic solution, toxic chemical, radioisotopes, vaccines and carbohydrates. Filtration is basically used in the heat sensitive products like carbohydrates, our macromolecules, our biological compounds are heat sensitive. They get denatured when we expose them to heat. So a filter is made of million of pores which only allow the passage of liquid or gas through them. These pores are smaller than the microorganism, hence it traps those particles which are larger than the pores. A filter is made up of so many pores. If the particle size is bigger than the pore size, it won't pass through it. It will retentate over it. There are present different types of filter that are used for different purposes. Each of them are made of different materials. So we have different types of filters. They are made of different types of material based on their property. Like it can be made from borosilicate glass, cellulose, sintered glass. Membrane filtration. In biology laboratories, membrane filtration is widely used to sterilize liquid. It is known as molecular or biological filter. High tensile strength polymers such as cellulose acetate, cellulose nitrate or polysulfonate are the main constituents of membrane filter. The contaminants which are larger than the pore size are trapped on the surface of the membrane filter. That's how they filter the liquid. If the desired particles are larger than the contaminant, then we can trap the desired particles or product on the membrane filter by decreasing the pore size, while the contaminants will pass through the membrane. I have told you that if the pore size is bigger than the desired particles, then it will go down. And if in case our desired particle size is bigger than the pore size, then what we will do, we will trap them on the filter and the contaminants will go down. Membrane can also be hydrophilic or hydrophobic. Hydrophilic is the water attracting and hydrophobic is a water repelling. Microfilter and ultrafilter membranes may be constructed from a wide variety of materials including cellulose acetate, polysulfonate, other polymers. Each of these material has a different properties with respect to the surface charge, degree of hydrophobicity, pH and oxidant tolerance, strength and flexibility. Membranes with a particular surface charge may remove particulate or microbial contaminants of the opposite charge due to electrostatic attraction. So here are some classification of membranes. If we talk about on the basis of material, material can be biological or synthetic. If it's biological, then it's a cell membrane. If it's synthetic, it can be made from organic polymers or inorganic polymers or hybrid. Organic polymers are glassy and rubbery. Inorganic are ceramics, glass, metals, and zeolites. If we talk about on the basis of structure, it can be symmetric and asymmetric. Symmetric are homogeneous films, cylindrical pores, and sponge-like pores. In asymmetric, integral asymmetric or composite. Integral asymmetric can be porous skin layers, homogeneous skin layers, and homogeneous skin layer. Or on the basis of membrane configuration, there are so many types of membrane configuration: flat sheet, spiral wound, tubular, capillary, and hollow fiber. So here are some applications of membrane filter: air protection, water protection, pharmacy, medicine, food, soil protection, agriculture, breeding, and energetics. So there are present many four types of membrane filters. These filters are classified based on their pore size: microfiltration, ultrafiltration, nanofiltration, and reverse osmosis. Here are the some comparison of the membrane filter. If we look at the diagram, you can see from the microfiltration to reverse osmosis, pore size is getting smaller and the pressure bar is increasing, and the permeate microfiltration will permeate water, low molecular weight solute, and colloids pass through them. Ultrafiltration will permeate water, low molecular weight solute, and nanomolecules. Nanofiltration will allow water, low molecular weight solutes, and reverse osmosis will allow only water. So from here we can understand this microfiltration. The pore size is 0.1 micrometer and it will separate bacteria, algae, sediments, and particles. Ultrafiltration, the pore size is 0.01 micrometer pore and it separates small colloids virus. Nanofiltration, pore size is 0.001 micrometer pores and it separates dissolved organic matter, divalent ions like calcium and magnesium. Reverse osmosis, it will separate NaCl monovalent ions and it will pass the water. So from microfiltration and ultrafiltration, it requires low pressure, and nanofiltration and reverse osmosis, it requires high pressure. So microfiltration, we filter at the microbe level. We can filter actual the microbes. So let's see. It separates those particles that have a size range of 0.1 to 10 micrometer. From the approximate molecular weight, it separates those macromolecules which has a molecular weight of less than 1 lakh gram per mole. It is designed to separate sediments, algae, protozoa, or large bacteria from the supplied liquid sample. It is a pre-treatment process of ultrafiltration and post-treatment process for granular media filtration. So here are some applications of microfiltration: fat, microbial removal in whey protein, isolate, casein whey fractionation, clarification of fermentation broth, microbial removal clarification, plant extract clarification, gelatin clarification, wine clarification, corn wet milling, industrial process wastewater treatment. Whey protein, I think we all are aware of whey protein. Whey is the watery residual left when we make paneer at home. When we make paneer at home, we see the residual left behind that that watery substance. It contains whey protein. Whey protein boosts the muscle growth. And here is ultrafiltration. It acts as a filter and separation barrier between two sites with a pressure difference low and high. It is the difference in pressure which acts as the driving force for solute smaller than the pore size to pass through a high to low pressure environment, trapping the load solute of waste. Ultrafiltration can effectively retain and separate salts, proteins, enzymes, gelatins, colloidal silica, manganese, iron, total organic carbon, bacteria, viruses, and chlorine resistant pathogens from water. So you can see the working. Here we will apply the pressure. We will apply the pressure and it will create a pressure difference. Then the pressure will allow the particles to move through the semi permeable membrane. The particles which have passed through the semi permeable membrane are the permeate, and which left over the membrane are retentate. The relationship between the applied pressure on the solution to be separated and the flux through the membrane is commonly described by the Darcy equation. We have a Darcy equation which explains the relationship between pressure on the solution and separated flux. So J is the flux is equal to TMP by mu RT, where G is the flux, TMP is the transmembrane pressure. It is the pressure difference between feed and permeate stream. Mu is solvent viscosity, and RT is the total resistance offered by the membrane and fouling resistance. So here you can see the membrane of ultrafiltration. The particles are here, the blocked particles, and over the membrane. The membrane used for ultrafiltration should have high porosity, narrow pore size distribution. High porosity means in a particular area there should be large number of pores. Factor affecting retentivity of membrane. There are some factors which affect the retentivity of membrane, जैसे कि size of molecules. If the molecule size is bigger than the pore size, it will retentate over the membrane, and if the molecule size is smaller than the pore size, it will pass through it. Shape of the molecule. If the shape of the molecule is different from the pore shape, it will retentate over it. Membrane material. We choose the membrane material according to our solution which we want to filter. Presence of other solutes. Membrane configuration. Membrane configuration, we have discussed so many types of membrane configuration, tubular, sheet, like, and fouling and adsorption effects. Fouling is the accumulation of the retentate over the membrane and between the pores, so it clogs the pores. The major challenge faced in ultrafiltration is membrane fouling. Over time, the filtrate will accumulate on the membrane surface and clog the pore. It greatly reduces the membrane effectiveness and efficiency. If the pores get clogged, it will reduce the membrane effectiveness and efficiency. Obviously, the particles will not pass through them. This reduces the effective TMP of the system, therefore reducing permeation rate. The increase in concentrate layer at the membrane wall decrease the permeate flux due to increase in resistance, which reduce the driving force for solvent to transport through membrane surface. Methods have been developed to reduce this effect for continuous operations. And here are some methods to reduce membrane fouling. By optimize pH and ionic strength of the feed solution to minimize the adsorption or deposition of the feed materials. By selecting an appropriate pre-filtration processor or other means to remove large molecules. Select a membrane with optimum pore size. Optimize the operating conditions. This includes increasing transmembrane pressure to maximize flux. Here are some applications. I know you are wondering, these applications are same as the microfiltration, but you know what, why? Because when we want to filter anything, we have to filter that thing from so many types of membrane, like first from microfiltration, then from ultrafiltration, then from nanofiltration, and then it goes to reverse osmosis. So here are the same: fat microbial removal in whey protein, casein whey fractionation, clarification or fermentation of broths, microbial removal, plant extract clarification, gelatin clarification, wine clarification, corn wet milling, and industrial process waste treatment. So you can see from this diagram, the red arrow shows that wastewater, and we will call it feed. It goes down, and here the white particles, I hope you can see, there are the pores. This is the filter, and the clean water is coming out of it. the particles like bacteria and viruses got trapped on the filter and the water passed through it. So, like, see, milk processing with membranes. We are all aware of milk products. We get so many different products from a milk. So, let's see how we get them. If we will pass milk from the membrane microfiltration membrane, we will get the cream. We will filter it low bacteria, low fat milk. Okay, from ultrafiltration, if the retentate will help in cheese manufacturing, specialty milk products, total protein isolates, and the permeate will lactose manufacturer, or it will help in fermented products. And then it will go down from the RO. The RO concentrate will be the evaporated products, dried milk products, bulk milk transporter, or specialty products, and the permeate will be the water. So another example is water treatment. The you can see the water is passing through so different types of membranes, and then in the end we got the permeate, that means the clear water. So first the our raw water will pass through the microfiltration. From the microfiltration, the retentate will be the suspended particles, oil emulsions, bacteria cells, and colloidal haze. And then it will pass through the microfiltration. The permeate will pass through the microfiltration. Then it will go to the ultrafiltration. The viruses, macromolecules, protein, and some molecular organic groups will retentate over ultrafiltration membrane, and the residual permeate will pass through it. Then it goes to the nanofiltration. Here some molecular organic groups and divalents will be retentate, and the residual permeate will pass through it. And then it will go to the reverse osmosis, where divalent monovalent ions will accumulate and the water will pass through it, and we get the pure water. So here are some disadvantages of membrane filtration. The turbid water cannot be used in membrane filtration. There may be a risk of bacterial abundance as the water carries numerous microorganisms. Glass filters are breakable and can break quickly. The membrane filters can crack easily. Only liquids are sterilized by this method. Filters are costly to repair, mainly nanofilters. Constitutional restrictions of supplies used in filters alter the effectiveness of this process, such as damage of glass filters, fracture of the membrane filter, and consumption of the filtrate by SIDS filter. It requires a high differential pressure, and clogging can occur. So here are some references from where I have collected this content. And in the last, I would like to thank you all. Thank you so much.